So our first speaker is Dr. John Bucher. He's Associate Director of the National Toxicology Program. In particular, his research interests are characterizing the toxicity and carcinogenicity of chemicals that are of interest to the NTP. He's responsible for oversight of the NTP toxicology and carcinogenesis studies, the NTP report on carcinogens, and the NTP Office of Health and Translation. He's spent a great deal of time thinking about toxicity testing, how to make it more efficient, better, and faster. And Dr. Bucher is going to talk to us today about testing methods for low-dose toxicity, including new in vivo and in vitro methods. Thanks.
Thank you very much. It's a pleasure to be asked to speak to you today. I have kept the title that I was given. I'm not so sure that's something that I should be thankful for because it's quite a large topic, but I'm going to try to do as best I can. I thought I would talk a little bit, just very briefly, describe what we are, talk a little bit about traditional assays, give you a response that we've made to a data poor emergency situation, talk about the low-dose issues with BPA, and then some alternatives approach. This really is going to not directly address the topic that was given to me on the by the title, but I'm going to come at it from, I think, 3 different directions. And maybe once you see this, you'll see why I've done it this way. So the National Toxicology Program is part of the National Institutes of Health. We also have collaborations at CDC and FDA, and we basically have, for the last almost 40 years, been doing studies on nominated substances to us for characterization of the toxicity and carcinogenicity of these agents. We also are responsible for some analysis activities, which you just heard. So we all know that there's very diverse kinds of information that can be used to make public health decisions. And all of these things you are probably aware of in various shapes and forms. But I'd also like to point out that there are also methodologies being developed now by our program and others to be able to pull the information together from these various data streams in a systematic manner and in really pulling together systematic review methods and tools to evaluate and integrate the findings from a wide variety of different information databases. So the standard NTP toxicology studies that we've been doing for many, many years are kind of listed here. I'm not going to go through these, but those of you who are familiar with these kinds of assays will appreciate that A, they take a long time, B, they take a lot of animals generally, and C, they cost a lot of money. None of those things are optimal for making decisions about green chemistry or new can linings or information in which you need answers fairly quickly. So I'm going to tell you a story about something that happened in Charleston, West Virginia. You may remember this back in 2014, where we had a data poor emergency situation in which the residents noticed a sweet smell that became quite pungent in the air. And what had happened was that there was a leak of about 10,000 gallons of material from tanks on the Elk River, which was about 1.5 miles upstream from the water intake for the water distribution system for the Charleston area, serving about 300,000 people. The tank material was used to clean coal, and it was primarily something called methylcyclohexane methanol, which is the starred chemical on your left. There were a number of byproducts present, which is somewhat similar to manufacturing new can linings with new contaminants. And there were a couple of structurally unrelated phenyl ethers there in much lower concentrations. So within about 2 days, the Centers for Disease Control was asked to come up with a permissible number. And this was for the amount of this material that could be safely consumed in drinking water. So they did the usual. There was one study from Eastman Chemical, which was the manufacturer of the chemicals. They had what they called a point of departure, or a lowest effect level, in their 28-day rat study, and then there were safety factors applied, and they came up with what they call a drinking water advisory level of 1 part per million. So the drinking water actually contained at the peak about 3 to 4 parts per million, but this was a transient exposure. It only lasted for a couple of days. Most people weren't drinking the water, but there were still some uncertainties about this situation. Very few toxicology studies that were available to support this drinking water advisory level. There was no information in developing animals. There were a lot of pregnant women in Charleston, as there are, and that was a concern. There are very limited data on the minor components of the spill. In essence, nothing really. So what we were asked to do by the senator from West Virginia and some other folks was to come up with a series of studies that might allow us to give some confidence to the drinking water advisory level that was set by the CDC. So we came up with a series of studies, including some rapid predictive screens, including structure-activity relationships, high throughput screens, the Tox21 program that was nicely already described, so I don't have to go through that in any detail. But this uses human cells for gene expression changes and other indicators of response in pathways of toxicological concern. So roboticized methodology. We also looked at C. elegans toxicity, where you expose these nematodes for effects and look at the effects on reproduction and growth and behavior. We looked at zebrafish embryo toxicity to monitor for effects on structural and functional development of zebrafish. We did some genetic toxicity assays, including the Ames test and a few others. We also did some rodent studies, and these, of course, took longer, but were, I think, very interesting because we've been sort of experimenting, if you will, with something that we call a 5-day toxicogenomics study. And instead of looking for effects in this assay, what we're looking for is actually levels of exposure that don't cause effects. So in this study, chemicals are given to rats for 5 days, and we took the livers and the kidneys, and we assayed them for evidence of changes in the expression of genes that were known to be associated with responses to toxic chemicals. So basically, you're doing dose response, and you're looking for things where the exposures don't happen. And again, what we're looking for here is to try to develop confidence in this 1 part per million drinking water advisory level. We also did some traditional assays, mouse dermal irritation and hypersensitivity studies. These are very irritating chemicals. The populace reported nausea, irritation, and skin irritation and things of that nature. And we also did rat prenatal toxicity studies to determine the effects on offspring of pregnant rats that received the chemical during pregnancy. So this 5-day toxicogenomics study I'm going to just talk briefly a little bit about. We used 6 dose levels from 0.1 to 500 milligrams per kilogram. Again, we measured liver and kidney global gene expressions, and we determined what is called the most sensitive molecular biological process, which was a group of genes that functioned together to control a particular cellular process. We run benchmark dose software on these things. Sorry if it's a way of determining where the dose response really rises out of the uncertainty, and you can actually measure that something is really going on. And you identify, in essence, a biological no effect level, which in our hands and in the hands of others, this typically occurs at a dose within a factor of 10 below that, which will cause phenotypic changes in more traditional studies. So I won't tell you about all the results, but I'll put the results that we came up with in the context of the study goals that we had, the first one was to strengthen the science base. We had SAR predictions with moderate confidence that these chemicals might cause developmental toxicity and irritancy, and in fact, those were in fact confirmed in our studies. In our rat prenatal toxicity studies, we came up with about the same kind of dose response for a low effect level in our studies with respect to the 28-day Eastman Chemical Company studies that were used to set the drinking water advisory level. In the 5-day toxicogenomic studies, these molecular biological processes were activated in fact at approximately 10-fold lower doses than phenotypic changes that had been reported in other studies. We found that the materials were less irritating and less sensitizing than they would have appeared to have been to humans in our animal studies. And there was very low genotoxicity potential, minimizing the concern for long-term health effects. With respect to sensitive life stages, the major components of the spill did not affect either the C. elegans or the zebrafish development. In our rat prenatal studies, though, we did notice lower birth weights in animals in the offspring, and these occurred at lower doses than where there was overt toxicity in the pregnant animals. However, the toxicity occurred at far above the drinking water advisory level. But it was interesting because this low birth weight was picked up by the state of West Virginia, and they did a birth weight study the year before the spill and the year following the spill to see if, in fact, there was any way that there might have been an effect on this endpoint, and this was negative. So with respect to the minor components, generally there were minimal differences in the minor components and the major methyl cyclohexane methanol. One of these minor components was more toxic to developing zebrafish and was mutagenic. So overall, we felt that the collected findings supported the adequacy of the drinking water advisory level. So that's one story. That's a data poor situation where you basically throw a lot of things, a lot of possibilities at it, and to basically increase your confidence in a situation. So there's been a conceptual shift in environmental health science, and this relates to the title of issuing the issue of low-dose toxicity. The old theories of chemicals acting by overwhelming the body's defenses by brute force at very high doses, not necessarily the case anymore, as more and more we learn about chemicals acting like hormones and drugs to disrupt development and function at very low doses to which the average person may be exposed. So, and the idea here is that these low-dose exposures during development can can induce perhaps disease later in life and potentially across generations. Now, the focus on bisphenol A is one, obviously we're not here to talk about the toxicity of bisphenol A, but I will say that you all know that there's still a very active controversy about the actual levels at which bisphenol A might be causing biological effects. If you look at the regulatory guideline studies that were used to set many of the permissible exposure levels, you'll find that they're set based at fairly high levels, where the endpoints that were seen in those studies have been minor, I would say, in comparison with the endpoints that have been described in many academic studies looking at much, much lower doses in animals and also in epidemiology studies where effects on a variety of different things have been shown in relation to ambient, if you will, exposures to bisphenol A. So we tried to put together a series of studies that tried to marry these two issues. So we at the Food and Drug Administration, we've actually in their Arkansas facility, we've actually comprised a very, very large GLP perinatal 2-year study with a wide dose range of bisphenol A to try to mimic both the very, very low doses and the very, very high doses that are seen in the guideline studies and have also been used in academic studies. We have carried out this study, and we also involved a number of different investigators who have, in their own hands, in their own laboratories, have generated information that would suggest that in fact bisphenol A was acting at very, very low doses. So we've asked them to basically take the materials from this guideline study and apply it in their own laboratories, analyze them using their own processes, and see how the results compare. We're not finished with these studies yet, but I will, just to give you an indication, I'm not going to go through these in any detail, but you can see the kind of endpoints that people have looked at in the bisphenol A research programs in their own laboratories. And these are the investigators. And you'll notice as you read through these that they are not the kind of endpoints that are typically looked for in the guideline and regulatory toxicology studies. And they often focus more on the molecular aspects of the assays systems that they're looking at, more so than an actual functional or a phenotypic outcome in a toxicology study. So there are a lot of these. And this is a very, very comprehensive study, and we're still in the process of, all of the measurements have been done by these people, but we're in the process of pulling all this information together. These studies were all done blinded. All the samples were sent to the investigators in a blind fashion. They were required to send all of their raw data to the NTP, and we would not decode their data until they had sent all of their data to us. So then the data went back to them, and they're carrying out their analyses, knowing the dose levels, and now writing their papers. So that's the second story, and I gave you that story because it illustrates the issue of where we are with respect to understanding low-dose effects in terms of long-term health effects, even for a chemical that's been studied as intensively as bisphenol A. For, as you mentioned, 10,000 publications, and there's probably 11,000 by the end of the day. So, getting back to the topic, the third topic I wanted to briefly talk about is what's the biological activity of the BPA analogues of emerging public health concern? And this is a project that one of our postdocs, Katie Pilch, put together. And it's basically has a number of different facets to it, working with other aspects of our program, doing very, very systematic literature reviews, looking at the high throughput screening data from the Tox21 and the ToxCast programs, looking at a number of short-term and long-term studies to begin to understand the relationships between bisphenol A and these various structural analogs. What are we looking at? Well, these are the chemicals that Katie pulled out of the literature and has been studying over the past couple of years. The ones in red are known or potential use in thermal paper. We had a study out. We're some of the first to show the substitution patterns with some, in fact, new chemicals that we didn't even expect to see in thermal paper showing up. The blue have been detected in environmental or in human samples. Many of these have been in human samples. Some of them are coming from, we believe, from food can linings and other food sources. The green, flame retardants. The orange, plastic resins and dental polymers. So they have a wide variety of uses, and they have a wide variety of potential places where they may be coming from. So I mentioned the systematic review methodologies before because they're becoming more and more important in trying to understand exactly where we should put our time and effort in designing studies to look at these things. We now have text mining tools using machine learning that will allow us to aggregate very, very large databases of information and allow a person to begin to go through looking at title and abstract and trying to decide whether that particular piece of the literature is of interest. And every time one picks a paper, the software will completely reorder the other 50,000 references such that it learns what you're interested in as you go along. So it's fascinating information. It's going to help, I think, in many respects in developing new processes for looking at alternatives. And of course this includes gray literature, the ECHO REACH database, and ToxCast and Tox21 high throughput screening. And there are a number of systematic review tools that have been developed recently, and here are just a couple, some of which are commercial and some of which are free. So the data streams, integrating the data. We need to pull together animal and in vitro data from all these literature searches. As I said, the ECHO and REACH databases, there are a number of hazard IDs that have been developed for the US EPA, which is now the Safer Chemicals Program, and the high throughput screening data. And if you look at that, you can just look at the inventory of available literature, and you'll see that there are an awful lot of zeros here. There are only 4 human studies related to all these different chemicals. A very small number of animal studies, a larger number of in vitro studies, but an awful lot of things are on the market with really nothing available. And there are no records at all for this set on the right. So the Tox21 high throughput screening capabilities have been described earlier. So we're not going to have to do that, but this is a picture of at the National Center for Advancing Translational Sciences, where we carry out our Tox21 screening program. This is a robotic activity. They have remarkable compound handling capabilities and informatic tools, and we've been screening over 8,000 compounds against, it's now about 75 nuclear receptors and stress response pathways using cell-based assays. And we're moving towards transcriptomic assessments. And I say this because it harkens back to the 5-day toxicogenomic studies in rats, where basically we're doing the same thing in the transcriptomic studies. We've developed the capability to be able to look at global gene expression in cells in a well and be able to put together these molecular biological processes so that we can understand the dose response of these gene changes in response to a particular chemical. These are the kind of data that we've been generating out of the Tox21 program with a single endpoint output, like simple like an estrogen receptor alpha. And to give you an idea of how reproducible these data are and how you can calculate EC50s or AC50s pretty well. You can also take this kind of information and display it in ways that allow you to really compare the studies, compare the various analogs with respect to their outputs. So on the right of this slide, you can see a number of different of the Tox21 assay endpoints, and they're all keyed. And on the bottom of this slide are the various substances in that list of substances that Katie pulled out that have been assayed in the Tox21 program. And then they're all arrayed across the micromolar dose at which they activate these various assays. So that's one way of looking at it. You can kind of see the patterns are emerging in the data. There's another nice tool the EPA put together, which is called a ToxPy. And basically what you do in this instance is you just array the information and in essence, the response in all these various assays in a pie-shaped form. And then you can simply visually look at these across the bottom of the slide and see how they relate to the E2, which is ethinyl estradiol, for example. And another way of doing this, which is a very interesting way to think about it as well, is that you can also model the chemical similarity between all of these various analogs and array them against the structural similarity. And it's kind of hard to see these here but in some instances, but basically bisphenol A is what you're comparing against. So that's 100% in chemical similarity and 100% in biological similarity and 100% in structural similarity. And then the other ones sort of array out with respect to their other activity profiles, and they get further and further away from bisphenol A. So I think there's a lot of new tools and things that are coming on the horizon that will help us understand how to move forward in selecting, making good choices with respect to green chemistry and the alternatives that are going to be most promising and most useful. So the conclusions, we have a wide variety of approaches to assess potential toxicity. Many are simply too expensive and too time-consuming to use in a screening method. We have a lot of screening assays. The low-dose toxicity that I will tell you that most of the academic investigators are finding is mainly at the molecular level, more so than at the phenotypic level. We don't know if that's really the thing we need to be worried about. I have a tendency to believe that that is something we need to worry about. But again, the translation of these low-dose effects to traditional toxicity endpoints is under active investigation. And we need to think more about toxicology with respect to finding doses and finding exposures and using all of the ExpoCast information to understand the exposures that are not going to cause something. If we can be assured that, in fact, if you don't have a response in a particular cell, if that organ simply isn't responding in any way that we can see with any kind of expression of gene changes, we need to worry about it. And this is still something that we're actively investigating and is going to be a large part of our research program over the next couple of years. So I think that's the end of my talk. I'm almost on time.